how the 4D nucleome, or 3D nucleome plus time, responds to DNA damage. And so the title, which I sort of somewhat modified since the draft, was Perturbing the 4D nucleome with DNA Damage on Demand. So we're, of course, all interested in the 4D nucleome. And usually to study systems like this one, a standard way would be to first perturb the system and then observe the response, right? And one such perturbation we can make is DNA damage, and more specifically, double-strand breaks. So double-strand breaks are important and interesting because although they are, as you can probably assume, pretty toxic to the cell, they're actually very common and can occur both naturally or as a product of external perturbations. Now, ways double-strand breaks can occur inside cells include application of genotoxic agents such as for chemotherapy, or X-rays, free radical damage, replication errors, and more recently, as a targeted induction using sequence-specific endonucleases like Cas9, CRISPR-Cas9, to cause genome editing. But to better study double-strand breaks and the cellular response, we want a good way to induce it on demand in both space in 3D as well as over time. Now, there are currently some methods to induce double-strand breaks, but they have a couple limitations. Some include physical or chemical mutagens, right, which can efficiently, shown here, and quickly cause DNA damage, but results in mixed lesions without control over the specific position. On the other hand, there are inducible endonucleases like homing endonucleases or an inducible CRISPR-Cas9 system, which can cause target-specific double-strand breaks, but previously have somewhat reduced kinetics and efficiency. So we hypothesized that if we could have an inducible Cas9 system, but with much faster kinetics and higher efficiency, then the system could be optimal to study the DNA damage response. And the approach that we came up with is something that goes along like this. So here's just an analogy. So you can imagine Cas9 as scissors parked on genomic DNA, shown here. But these scissors can't cut because you have a rock here that blocks the cleavage action, of course. But then just imagine for a moment that if you can shine light, and then that can just evict the rock, then these scissors can immediately start cutting DNA. And molecularly, what that corresponds to is you have Cas9 with a CRISPR RNA that contains distal caging groups that can block the binding of this CRISPR RNA to the genomic DNA shown here. So Cas9 can still bind to genomic DNA, but it cannot cleave because these blocking groups prevent full hybridization. Upon a brief exposure to light, however, these distal caging groups are actually quickly released, and which allows full complementarity between the DNA and the Cas9 guide RNA, which activates Cas9 to cleave target DNA. So here are just some preliminary results. To measure the speed of Cas9 activation inside cells, we can measure the percent of broken DNA at the target site, shown on the y-axis, as a function of time after Cas9 activation or on the x-axis. And our method, in the red line, is compared to a prior state-of-the-art chemically inducible Cas9. And it performs better in two distinct ways. First, our method achieves, our method is orders of magnitude faster, right? So reaching over 50% of double-strand breaks within 30 seconds of Cas9 activation. And second, our method intrinsically has higher cleavage activity, consistently having a higher percentage of breaks over all time points. Let me just switch to a laser pointer here. Perfect. And so given these qualities, we can call our method very fast CRISPR. Now we can use this strategy, this fast and efficient strategy for DNA damage induction, which I just presented, to really study how the 4D nucleome responds to DNA damage. And we can perform the following experiment. So we first deliver Cas9 into cells and then allow stable Cas9 binding. And then we can shine light to activate Cas9 simultaneously within a cell population. And then we can harvest these cells for subsequent analysis as a function of time after activation. The readout we first use is something called ChIP-seq. And in ChIP-seq, we're interested in where our protein of interest binds genome-wide. To determine this, we use antibodies to first pull down our protein of interest, and then sequence the DNA that's attached to the protein. And finally, computationally determine where these DNA sequences actually map to the genome. So first, chromatin immunoprecipitation followed by sequencing. And by combining ChIP-seq with our very fast CRISPR system, we can first study how the cell responds to Cas9-mediated DNA damage. The first, we look at MRE11. So MRE11 is a protein believed to initially find and bind DNA breaks. And after performing ChIP-seq for MRE11, and these are the results we obtained, and the results are shown in a 2 kilobase window centered at the Cas9 cut site. And each row here corresponds to a different time point after Cas9 activation, from 0 minutes, so no light, all the way to 1 hour after activation. And enrichment analysis determined that actually these double-strand breaks are detected by cells as early as 2 minutes after Cas9 cleavage, which is actually much faster than previously believed, because people used to think that after Cas9 cleavage, the Cas9 just stays bound to the genomic DNA and prevents the cell from actually detecting it in the first place. We can also measure how quickly the DNA damage response, specifically damage-induced histone phosphorylation, for example, spreads from the cut site. And to do this, we perform ChIP-seq for damage-induced histone phosphorylation as a function of time. So this is gamma H2AX. And we measure the enrichment width at each time point, and we can determine the speed of its bidirectional spreading, shown here, to be approximately 2.5 kilobases per second. And interestingly, this speed is consistent with the speed of loop extrusion. And a recent report actually proposed histone phosphorylation, so this process to occur through bidirectional loop extrusion from around the cut site. So this was just a somewhat brief introduction to our prior work, which has been published already. But something we thought about when we were performing this is we were always looking at the Cas9 double-strand break response at one unique sequence, right? But suppose for a second, what if that sequence were located somewhere else in the genome? Would the response change at all? And if so, how would it actually change? But to answer this question, ideally we would have a one Cas9 guide RNA sequence that could not target one site, but could target many places in the genome, either from tens to hundreds or maybe even more. And then the behavior at each target position can be determined and then correlated back to its genomic position. So to accomplish this, we first developed a pipeline to really discover, well, to first know if these guide RNAs, so these Cas9 target sequences, even exist. And if so, their identities. And luckily for us, we found that they do exist. We were able to identify over 75,000 of them, and each can target between 1 to over 1,000 genome-wide target positions. Furthermore, we found that they actually target epigenetically diverse positions, ranging from enhancers to transcribed regions or to quiescent or repressed regions as well. Now we focus on a subset of these guide RNA sequences. And just to remember, each of these sequences can target, we focus on between 100 to 300 positions genome-wide. And one important point here is we selectively find these guide RNAs which target positions that can be uniquely distinguished using sequencing, because we want to accurately map, for example, the enrichment of repair factors or other readouts around these target positions. So for the rest of the study, I focus on one particular guide RNA sequence, which targets around 126 positions genome-wide, but have over 4,000 detected binding sites if you don't have any cleavage. So for this experiment, we first deliver, as I mentioned, one such multi-target guide RNA sequence in complex with Cas9. And in this case, we use cleavage deficient Cas9, so dCas9, to solely measure Cas9 binding without any effect from cleavage. And so across these over 4,000 detected Cas9 binding sites, many of them, of course, have mismatches between genomic DNA and your Cas9 guide RNA. In this plot, the y-axis shows the level of Cas9 binding for each target position, grouped by the type of mismatch along the x-axis. So 0, 0 here indicates all target positions without any mismatches. And there's quite a lot going on here, but I just want to point our attention to the first row, where even for identical target positions with no mismatches, there's actually very wide variability in enrichment between target positions. So there's, as I mentioned, 126 such positions, and the enrichment of Cas9 at this time point, so 3 hours here, ranges from close to 0 to over 8,000, and you can just assume arbitrary units. And this suggests influences from other factors, like, for example, epigenetics. We also do the same experiment, but with active Cas9, and then follow that up with MRE11 ChIP-seq to measure the Cas9-induced DNA damage response. And we again observe this very wide variability if you just focus on the first row here in enrichment of MRE11. Now, this raises the question, of course, of what can explain this observed heterogeneity between identical target sequences, but which happen to be found in different genomic locations. And to answer this question, we analyzed 9 publicly available epigenetic data sets from ENCODE. And for those who don't really know what these correspond to, they correspond to regions of either high accessibility, or they mark enhancer regions, gene bodies, promoters, places with nucleosomes, or inactive chromatin as well. And then we can correlate, for example, the level of Cas9 binding to these epigenetic markers at all Cas9 target positions. And what we found is, if you look at Cas9 binding, so without any cleavage, we found that Cas9 binding is most correlated to chromatin accessibility, mostly, for example, DNA-seq and ATAC-seq. And this is actually pretty consistent with prior literature, which showed that Cas9 binds better at chromatin accessible regions. So this further validates the correctness of our analysis. But interestingly, if you look at the MRE shown here after active Cas9, the correlation, there's other stuff going on here, right? And to figure out what exactly is causing these extra correlations outside of increased accessibility from Cas9 binding, we actually normalize the MRE11 damage response by Cas9 binding. So we normalize out Cas9 binding and focus only on what is causing differential MRE11 damage response. And interestingly, we found the highest correlation actually to gene markers, for example, H3K36 trimethylation and RNA-seq, which of course maps to transcribed regions. So in summary, we found where we confirmed that Cas9 binding is preferential to accessible chromatin, and also Cas9 cleavage is interestingly, there's higher levels actually of Cas9 cleavage near genes and promoter regions. And lastly, we found that these phenotypes are actually predictable using machine learning. And so I won't go into the models we used here, but we can achieve correlation coefficients between the expected and the predicted levels of enrichment with an independent test set of over 0.8. And so this is actually pretty highly correlated. And lastly, I'd like to focus on an additional question, which is how the cell responds. So how the cell's chromatin accessibility actually changes due to DNA damage. And to measure chromatin accessibility, we use ATAC-seq, which looks for regions where there's very accessible chromatin. And we look at the average ATAC-seq enrichment across all target sites in cells both exposed to Cas9 or without Cas9. And the x-axis plots the base pair distance away from the Cas9 cut sites. And what we see is very high accessibility centered at target positions only for cells exposed to Cas9. And we found that the region of accessibility is constrained to within 1 to 2 kilobases from the cut site. And I won't go into the results here, but what we found is that this accessibility is primarily nucleosome free. And just consistent with prior data that we showed, where increased accessibility occurs 1 to 2 kilobases from the cut site. And if we think about an average distance of approximately 200 base pairs between each nucleosome, this indicates that the increased accessibility occurs in a region that's occupied approximately with 5 nucleosomes on either side of the cut site. And lastly, I want to show something, some newer results, which can combine our very fast CRISPR technology with multi-target CRISPR to study at what timescale this increased chromatin accessibility occurs. And to do so, we use 3 different readouts to measure phenotypes at all multi-target Cas9 positions. So first, we use a method called BLISS, which measures double-strand breaks directly genome-wide before activation, so 0 minutes, or at 10 minutes or 30 minutes afterwards. And that is compared to MRE11 ChIP-seq as well as ATAC-seq. And our enrichment quantification determined that actually double-strand breaks are, of course, first to increase, right? And that is followed by the DNA MRE11 detection of this double-strand break. And then afterwards by increased accessibility, which only goes above basal levels, at least in our measurements, at the 30-minute time point. So the point here is just that Ziem's chromatin accessibility due to DNA damage occurs after the cellular detection. And finally, I think this work has potential to facilitate collaborations with the 40 Nucleome network of investigators. So as I showed in our presentation, we have the ability to perturb the 40 Nucleome using targeted double-strand breaks on demand. And of course, by demand, I mean in control of both time and space. So any number of positions and its position genome-wide. And these samples can be shipped to collaborating labs to study its induced cellular phenotypes using novel readouts that they develop. And we're actually starting a collaboration with Dr. Diao at Duke University. And we're actually currently shipping the samples to him. So in summary, we have developed new technologies to induce targeted double-strand breaks on demand in both time and space. And the goal here is to understand the effect of DNA damage on the 40 nucleome. So I'd like to thank everyone for listening. And first, I'd like to acknowledge TJ, of course, as part of the TJ's HiLab, as well as 2 postdocs who, first Yang, who really pioneered the very fast CRISPR work, and Alberto, who performed many of the ATAC-seq experiments I just presented, in addition to some collaborating labs and the MD PhD program, as well as the BME PhD program and my thesis committee. So thank you all for listening.